Let's start, madam. Madam, start the class.
Hello.
Start the class, madam.
Hello.
Start, madam.
Okay. Good morning, everybody. Students, you have learned about the internal structure of dicot stem last week, and today you are going to learn internal structure of monocot stem. And before going to the topic, and I want to tell you something important. When you look at the pictures, some parts such as xylem and sclerenchyma, they look dark pink in color, and remaining tissues look light green in color. And these are the pictures seen in compound microscope under high power. Then while making slides, we use a stain called safranin solution, which colors xylem and sclerenchyma, so they look dark pink in color, and remaining tissues don't take the color. So while drawing diagrams also, to show this differentiation, you have to use three different kinds of pencils, HB, 2H and 2B. 2B should be used for xylem and sclerenchyma, and 2H and HB should be used for other tissues. Then, while drawing, you should not draw by putting zeros in lines. You learn from your lecturers how to draw in your regular class. Then coming to the today's lesson, that is internal structure of monocot stem. Of course, monocots are the plants with seeds with a single cotyledon, and the root system is of fibrous type, and leaves with a parallel venation, and flowers are trimerous flowers. Then let us go to the actual slides. Before that, we have to learn objectives. Coming to the learning objectives, to understand the internal structure of monocot stem, that is, to study the internal structure of monocot stem, we have to take the cross section in the region of internodes. Actually, what is a node? A node is a place where a leaf arises, is a node, and the distance between two successive nodes is known as internode. And to know in the internodal region only, we have to take the cross section to study the internal structure of monocot stem, and to know the tissue differentiation in monocot stem, that is epidermal tissue, the outermost one, then ground tissue inside the epidermis, and vascular tissue, that is the central portion, and to find out the differences between monocot and dicot stem. So these are the objectives. Then coming to the anatomy of the monocot stem, internal components of monocot stem, 4 tissue systems are there. First one, dermal tissue system. Next one is hypodermal tissue system. And third one is ground tissue system. And fourth one is vascular system. So dermal tissue system is the outermost, which contains epidermis. In this picture, you can see clearly epidermis, the outermost layer, then hypodermal tissue system, the next to the epidermis, and ground tissue system. That remaining part fully is known as the ground tissue system, and vascular tissue system. So it comprises of xylem and phloem, no cambium in this monocot stem. The main difference from dicot stem is ground tissue is not differentiated into cortex and endodermis in monocot. Then coming to the dermal tissue system, first one, so composed of epidermis. Epidermis is single layer, that is, it is present only in one layer and made up of parenchymatous cells. And you can see it is represented in, it has taken safranin stain, so looking some dark pink in color. Then cells are compactly packed. That means without any space, they are compactly arranged. Then thick layer of cuticle is also present. And outside epidermis, you find cuticle, which prevents water loss. And cell wall is also silicified. That is due to the deposition of silica, the cell wall becomes somewhat tough. Then epidermis persists as long as the stem lives. That is, here no secondary growth is seen. So as long as actually these plants are annuals, we find epidermis intact, persistent. Then hairs such as trichomes, what you found in dicot stem are absent here. Trichomes are absent. And here we have to talk about that cell wall is silicified due to the silica deposition. Human beings cannot take as grass plants as their food. That is, coming to the dicotyledonous coriander, palak, then amaranthus, all these leaf vegetables are taken by humans. But grasses, human beings, they don't have the capacity of digesting these plants because due to the deposition of silica. Only animals, the two herbivores, because they have ruminant, they are known as ruminant animals. By regurgitation, they bring back the food again to the mouth and they digest. That is why we don't take any monocotyledonous plants as a food. Then outside cuticle, you can see, which is very clear with arrow mark, it is shown, and which prevents water loss. Then a few stomata are found here. And now here and there you see some interruptions in the epidermis. It is very clearly shown in the picture diagram. And coming to those are stomata. And of course, stomata are helpful in gaseous exchange, and also in transpiration they help. Transpiration is loss of excess of water through stomata from aerial parts of the plants into the atmosphere. Coming to the dermal tissue system functions, it is the limiting layer, that is outer boundary. Then it provides protection also, protection of inner parts. And silica deposition provide mechanical support. And sometimes these plants are not paddy, other plants, they grow tall. So they provide mechanical support. When they grow tall, there is danger of falling down. So the silica deposition makes strong. Then cuticle prevents the transpiration and stomata allow gaseous exchange. In this way, these are the functions of dermal tissue system. Then hypodermal tissue system. It is composed of hypodermis. So next to the epidermis, it is shown with an arrow mark, a dark pink in color, that is hypodermis, occupy immediately below the epidermis. Just below the epidermis, you find hypodermis, and cells are polygonal and compactly packed and multi-layered and sclerenchymatous. Then alternate patches of chlorenchyma may also be present. Then, of course, chlorenchyma means a parenchyma with chloroplast, and which is photosynthetic in function. Then below chlorenchyma, hypodermis is continuous. And here you see the peripheral vascular bundles, that is, which are towards the epidermis, are small in size, and these towards the center are big in size. And peripheral vascular bundles are embedded in hypodermis, and coming to the function, provide support and protection. Then ground tissue system. Ground tissue system consists of only cortex, and here also cortex, no differentiation of outer cortex, inner cortex, and endodermis, only ground tissue. And ground tissue is not differentiated, and cortex parenchymatous, it is parenchymatous. Coming to the parenchyma, they are thin-walled cells, and with intercellular spaces between cells, then with a large vacuole in the center. And here, if it contains chloroplast, it is photosynthetic in function. Otherwise, with the large vacuole and in the ground tissue, storage in function. The nutrients are stored in this parenchyma, that is in the ground tissue. Then cells loosely packed with intercellular spaces that you know. So in between the cells, the spaces are clearly seen. Then in aquatic monocots such as potamogeton, parenchyma is present. In dicots also you learnt about parenchyma. Because of parenchyma, the hydrophytes, that is um, the plants which live in water, uh, they become buoyant, that is they float on the surface of water. This potamogeton plant is nothing but aquatic weed. It is a weed in the ponds, so it contains parenchyma in the ground tissue. Then in some plants, central portion is hollow, oryza, and oryza means paddy, triticum means wheat. Here the plant, the central portion is hollow. Coming to the function, storage, because it mainly contains parenchyma storage and bears vascular bundles also. Then coming to the vascular tissue system, cambium is absent. In dicotyledonous plants, you find cambium, and here in monocots, cambium is absent. So this vascular bundle is described as closed vascular bundle, and vascular bundle composed of xylem and phloem. Then sometimes on seeing these vascular bundles, you can recollect something. So near electric poles and other danger symbol is shown like that. So these look like this. These vascular bundles look like danger symbol. Due to the position of the xylem and phloem, they look like that. And xylem is endark. Endark means metaxylem towards the periphery and protoxylem towards the center. Protoxylem arranged towards the interior, that is towards the center. Metaxylem arranged towards the exterior. And xylem elements are arranged in the shape of V, the letter V. Then metaxylem elements form the arms of V, and protoxylem elements occupy at the junction of V. So this is the position. See, xylem vessels are clearly seen in the shape of V. They are arranged. They are known as xylem lacuna, also very big in size, metaxylem. Then coming to the, again, vascular tissue system, metaxylem is composed of two large vessels, xylem vessels. So xylem actually, it is a complex tissue because it is a combination of several tissues. Then due to the deposition of lignin, it prevents water loss. Lignin is deposited in xylem and prevents leakage of water. If once water enters the xylem vessels, it cannot be leaked out. Then tracheids are there, elongated tubes for supply of water, for conduction of water. And fibers, they are long tubes with thick walls. And vessels are there, vessels long, cylindrical, cells are lignified. And xylem parenchyma is also there. Then so sometimes for vascular, here for vascular bundle sheath also is seen. Then metaxylem composed of two large vessels, I told you. Then metaxylem vessels are with pitted thickenings. Pitted means some holes like that. Then metaxylem tracheids present between metaxylem vessels. Two metaxylem vessels are there. In between you find tracheids. Then protoxylem is composed of few vessels, and protoxylem vessels are with annular or spiral thickening. Annular means ring-like, spiral means spring-like thickening is seen in these protoxylem vessels. And protoxylem elements fuse to form lysogenous cavity. Then this lysogenous cavity is nothing but a cavity. This cavity is called protoxylem lacuna. Lacuna means cavity only, and protoxylem cavity is absent in asparagus. Asparagus is also a monocotyledonous plant. In the gardens and in the pots it is grown. Then next one is phloem. Phloem is situated outer to xylem between the arms of the V. The xylem vessels are arranged in the form of V, and phloem is composed of sieve tubes and companion cells. Phloem parenchyma is absent. Coming to the sieve tubes, that is, they are perforated. Perforated means they contain some holes or the pores. As at home we use jalleda, they look like that. Then these sieve tubes or sieve cells are associated with companion cells just adjacent to the sieve tubes. And phloem fiber is also present, which is known as bast fiber. And actually in dicots also, in jute, that fiber is known as bast fiber. Then bundle sheath is sclerenchymatous. Here sclerenchymatous means bundle sheath is very strong, sclerenchymatous. In raw gava, can you eat green raw gava? See, you cannot eat because of this tissue, sclerenchyma, dead tissue. Due to the deposition of lignin, you cannot eat raw gava uh, unless it is ripened. You can eat ripened this gava very easily. Then it is tough and waterproof also. And it gives some mechanical support due to the presence of fibers and also sclerids. So they are very hard and tough. So this bundle sheath is absent in asparagus. It is also a monocot plant. So this is the about the structure of monocot stem. Now let us study the differences between dicot stem and monocot stem. And sometimes this question appears in IPE examination. So dicot stem, these epidermal hairs are present, known as trichomes. In monocot stem, such epidermal hairs are absent. Then in dicot stem, hypodermis is collenchymatous, whereas in monocot stem, the hypodermis is sclerenchymatous. And collenchymatous means, again, they are polygonal cells and the corners are thickened, they are oval in shape, then no intercellular spaces. Due to the deposition of pectin or cellulose, these dicot stems are collenchymatous, whereas monocot stems are sclerenchymatous. So endodermis is present in dicot stem. Endodermis is absent in monocot stem, and ground tissue is differentiated to stellar and extrastellar regions. So after epidermis, you find hypodermis, inner cortex, outer cortex, endodermis, like that. You see differentiation in dicot stem, whereas in monocot stem, there is no such differentiation. Then vascular bundles are linked in are limited in number. That means some 6 to 8 will be there, vascular bundles in dicot stem. Whereas in monocot stem, vascular bundles are numerous, that means many. Then vascular bundles are arranged as a broken ring. So not fully concentric rings, broken rings, because here and there you find medullary rays in dicot stem. So they are like broken rings. And vascular bundles are scattered here. The vascular bundles are scattered in the ground tissue. Scattered means that is, if you stand in one place, throw some stones or some mustard seeds, they fall here and there. In such a way, vascular bundles are scattered here. Then vascular bundles are conjoined, collateral, and open in dicot stem. Conjoined means xylem and phloem together. You find collateral means side by side they find. That open means because due to the presence of cambium, they are described as open. Then coming to the monocot stem, vascular bundles are conjoined, collateral, same as in dicot, but closed. Because cambium is absent, they are described as closed. And vascular bundles, here in dicot stem, cambium is present. In monocot stem, cambium is absent. So, coming to some more differences, dicot stem, protoxylem lacuna is absent. In monocot stem, protoxylem lacuna is present, and pericycle is present here, and pericycle is absent in monocot stem, and bundle cap is present in dicot stem, whereas bundle cap is absent in monocot stem, and bundle sheath is absent in dicot stem, and you find the sclerenchymatous bundle sheath in monocot stem. Then phloem parenchyma and phloem fibers present in phloem in dicot stem. In monocot stem, phloem parenchyma and phloem fibers absent in phloem. Then pith is present in dicot stem in the center next to below the vascular bundles. You find some parenchymatous tissue which is large. It is known as pith. Such pith is absent in monocot stem. Then medullary rays are present in dicot stem in between vascular bundles, and medullary rays are absent in monocot stem. Then dicot stem undergoes secondary thickening, whereas monocot stem there is no secondary thickening. That is due to the absence of cambium, no secondary growth is seen. Okay, now let us review again, recapitulate. So what we have learned now. It's beginning better. It's beginning better. Outline. Okay, let us recapitulate what we have learned in this class. Coming to the anatomy of monocot stem, I told you what are monocots, and the plants which contain seeds with single cotyledon are monocots. And coming to the internal structure, we find 4 tissue systems: dermal tissue system, hypodermal tissue system, ground tissue system, and vascular tissue system. And here, the ground tissue is not differentiated into cortex and endodermis in monocots. And as in dicots, in monocots also composed of epidermis, which is single layered and mostly parenchymatous and compactly packed without any intercellular spaces, and a thick layer of cuticle is present to prevent water loss, and cell wall is silicified, that is due to the deposition of silica. Then epidermis persists as long as the stem is there, and mostly these monocots are annual plants. They live for 1 year or 1 season. So the epidermis only, because there is no cambium, no secondary growth. Epidermis only becomes the outermost layer, and trichomes, that is unicellular or multicellular hairs, are absent. Then coming to the dermal tissue system, then and here few stomata are present. And coming to this dermal tissue system functions, this is a limiting layer and outer boundary, provides protection. It is a protective layer, and silica deposition provide mechanical support also. When cuticle prevents transpiration, due to transpiration there will be water loss. It can be avoided by cuticle. And stomata allow gaseous exchange. Then coming to the hypodermal tissue system, it composed of hypodermis, occupy immediately below the epidermis. Next to the epidermis, we find hypodermis. Cells are polygonal and compactly packed. Polygonal means having many sides. And multi-layered and sclerenchymatous. Multi-layered means many layered. Then alternate patches of chlorenchyma may be present. Then below the chlorenchyma, hypodermis is continuous. Then peripheral vascular bundles are embedded in hypodermis, and it provides support and protection also. And coming to the ground tissue, it composed of cortex only. And ground tissue is not differentiated into outer cortex, inner cortex, endodermis, as in dicots. Here, uniformly arranged parenchymatous tissue is present, and cortex is parenchymatous, and mainly it is storage in function, storage of nutrients. Cells are large, circular, and loosely packed in the intercellular spaces. Then aquatic monocots, potamogeton, parenchyma is present. Potamogeton is an aquatic weed. Weed means unwanted plants which grows in the ponds. Then the stem is parenchymatous, that is having gaps in between, and it makes the plant buoyant, that is to float on the surface of water. In some plants, central portion is hollow, that is in the case of oryza, that is paddy, and triticum, that is wheat. Then coming to the function of this ground tissue system, storage and bears vascular bundles. Then vascular tissue system, it composed of vascular bundles. Here, vascular bundles are numerous. Vascular bundles are numerous, that means many. Unlike dicotyledonous stems, and the arrangement is scattered in the cortex. Scattered means that is throughout, here and there they are found. Then if you stand in a position and throw stones, they thrown scattered in the same way. Here, the vascular bundles arrangement is scattered, and vascular bundles widely separated from each other, not in the form of ring or attached. And vascular bundles show size differences also. The vascular bundles towards periphery are smaller in size, and vascular bundles towards the inner region are larger. And vascular bundles are conjoined, collateral, and closed. Xylem and phloem found together and adjacent to one another. Then because cambium is absent, they are described as closed. Then cambium is absent, that is why they are described as closed. Then vascular bundle is composed of xylem and phloem. Xylem is endark. Endark means metaxylem towards periphery, and protoxylem towards the center. Then protoxylem is arranged towards the interior. Then xylem elements are arranged in the shape of V, letter V. Then metaxylem elements forms the arms of V, whereas protoxylem elements occupy the junction of the V. Then metaxylem is composed of two large vessels. Large means big, very big vessels are shown in the picture, you see. Those are metaxylem vessels, and they are with pitted thickening. Pitted means having some holes. Then metaxylem tracheids present between metaxylem vessels, and tracheids also elongated tubes for the supply of water, for conduction of water. And protoxylem composed of few vessels only, xylem vessels. And protoxylem vessels are with annular or spiral thickenings. That is, annular means ring-like thickening, spiral means spring-like. And protoxylem elements fuse to form lysigenous cavity. Some cavities are there known as lysigenous cavities, and this cavity is called protoxylem lacuna. Lacuna means cavity only. And protoxylem cavity absent in asparagus, that is a monocotyledonous plant. There protoxylem is absent. Then phloem is situated outer to the xylem, in between the two arms. Then phloem is composed of sieve tubes and companion cells. I told you already, sieve tubes, they are perforated. They are perforated. Perforated means having some holes. Then companion cells means just adjacent to the sieve cells are known as companion cells. And phloem parenchyma is absent here. And vascular bundle in most cases are surrounded by bundle sheath. And this bundle sheath is clear and chimatous here. That is, around the vascular bundle you find bundle sheath, and bundle sheath is absent in asparagus. Then coming to this is the structure of monocot stem. Then there are differences between dicot and monocot stem. And coming to the dicot stem, epidermal hairs are present here known as trichomes, whereas in monocot stem, epidermal hairs are absent. Then in dicot stem, hypodermis is differentiated and it is cholenchymatous, and here hypodermis is sclerenchymatous. Endodermis is present in dicot stem, then endodermis is absent in monocot stem, and ground tissue is differentiated as extrastellar and intrastellar. Extrastellar means outside the vascular bundles, that is outside the still. Then intrastellar means the vascular bundles, that is inside the endodermis. And ground tissue is not differentiated in monocot stem into outer cortex, inner cortex, and other stellar regions, endodermis, like that. Then coming to the dicot stem, vascular bundles are limited in number. In dicot stem, 6 to 8 will be there, and they are arranged in the form of a ring also. And whereas vascular bundles are numerous in monocot stem, whereas the smaller ones are towards periphery and bigger ones are towards the center. Then vascular bundles are arranged as broken ring. Why they are in the form of broken ring is in the middle, in between the vascular bundles, you find medullary rays in dicot stem, whereas here vascular bundles are numerous and they are scattered in the scattered manner they are present. Then vascular bundles are conjoined, collateral, and open at dicot stem. Conjoined means xylem and phloem together and adjacent to one another, and they are in the form of a they are open. Open means due to the presence of cambium, the vascular bundle becomes open. Then here vascular bundles are conjoined, collateral, and closed. That means cambium is absent in monocot stems, so they are described as closed. And in dicot stems, cambium is present, whereas in monocot stem, cambium is absent. Then let us continue the differences between dicot and monocot stems. And protoxylem lacuna is absent in dicot stems, and in monocot stem, protoxylem lacuna is present. And in dicot stem, pericycle is present, and in monocot stem, pericycle is absent. And in dicot stem, bundle cap is present. Then in monocot stem, bundle cap is absent. Then in dicot stem, bundle sheath is absent. And in monocot stem, bundle sheath present. And phloem parenchyma and phloem fibers are present in phloem in dicot stem, whereas in monocot stem, phloem parenchyma and phloem fibers are absent. Then in dicot stem, large pith is present in the center, whereas pith is absent in monocot stem. Then medullary rays are present in between the vascular bundles in dicot stem, whereas medullary rays are absent in monocot stem. Then in dicot stem, we see secondary thickening, that is secondary growth, that is due to the presence of cambium, and interfascicular cambium also develops and cuts off secondary phloem and secondary xylem, so they participate in secondary growth. In monocot stem, no cambium and no secondary thickening, and they are annuals only, so we don't find secondary growth. And this is the structure of monocot stem. And these are the differences between dicot stem and monocot stem. With this, I am going to close this session or class. Thank you all. Thank you very much. అసలు ఈరోజు ఏం దిక్కు దోచకుండా అసలు ఏం సరిగానే అనిపించట్లే నాకైతే. ఇది పని చేయకపోయి ఇవి ఏం వర్కులు పోతాయి. అది కూడా టైం వచ్చింది పైన 5 నిమిషాలు 7 నిమిషాలు ఉంది.
మేడం వాళ్ళు చెప్పారా మీటింగ్ అయిపోయిందని? చెప్పారా మీటింగ్ అయిపోయిందని?
మామూలుగా చెప్పాను గోయింగ్ టు క్లోజ్ అని.